Now let's talk about prokaryotes. What makes a prokaryote? The main distinction and classification of eukaryotes versus prokaryotes is the presence or absence of a nucleus. As we mentioned, the nucleus, and you probably learned before, nucleus is that big thing in the center which holds the DNA and all the other stuff in animal cells and plant cells and stuff. Prokaryotes, however, do not have this central nucleus in which the genetic information is found. Instead, the genetic information is just stored inside of the cell, like floating around in most cases. Another main difference between prokaryotes and eukaryotes is the presence of a cell wall. All prokaryotes have a cell wall, but only some eukaryotes have a cell wall. As you probably know, plants have cell walls, but animals like humans don't have cell walls. So remember, all prokaryotes, all bacteria, all archaea have cell walls, but not all eukaryotes have cell walls. Some don't. Prokaryotes also have no membrane-bound organelles. Remember, the membrane is the outside, right? And then the organelles, some of them are membrane-bound. Like, for example, the endoplasmic reticulum is considered membrane-bound, which prokaryotes don't have membrane-bound. This is just very simple. You can just memorize like this. Okay. DNA in prokaryotes, right? So the main differences. The differences in DNA and genetic storage are the main distinctions between the cell types, right? So as mentioned, eukaryotic DNA is found in the nucleus, but in prokaryotic, nucleus doesn't exist, so DNA simply exists in the cell, right? Furthermore, prokaryotic DNA is circular in shape compared to the linear eukaryotic DNA. You probably remember eukaryotic DNA is more like this, whereas prokaryotic DNA, some of you may know of if you took genetics, plasmids in certain bacteria are used in genetics, genetic research now. They're circular, and that allows us to like kind of cut them like this, and you'd be able to take that, like snip that part out. And that's like a big thing in genetics right now, in genetic engineering and genetics in general. Similarly, prokaryotic DNA has no introns or bound proteins. DNA has a lot of proteins on it, and it has introns, which are not essentially called the non-coding regions. When you turn the DNA into RNA, so you have this DNA. When you turn this into RNA, a lot of these would get kind of spliced out. So maybe this section and this section would get spliced out, and only this part, and this part would be kept in for the RNA, and that would actually be what is used to make the proteins. So some sections are non-coding sections in eukaryotic DNA called introns. Most prokaryotes don't have introns. There may be some exceptions, but I think a good general rule is they don't have introns. They just have all coding regions. Okay. Okay, and this is an example of what a bacteria cell can look like. So you can see here it has this cell wall, has these pilus, has the membrane inside this yellow thing inside the cell wall. It has the DNA right here, just floating around, not in a nucleus. Has these ribosomes, again, not membrane bound. Cytoplasm, just like everything else. The flagellum, which we'll get to later on, is very important for mobility, for movement of the bacteria. Okay. Next, okay. So binary fission. As prokaryotes are always unicellular organisms, meaning that they only exist as one cell, whereas obviously, you know, humans and animals and plants are made up of many cells, and they're called multicellular. Unicellular organisms reproduce through a process called binary fission. The process is fairly similar to eukaryotic mitosis, but it's much simpler. There are many in mitosis. There are many stages and checkpoints, and different, like the mitotic spindle forms throughout mitosis. That's really there to ensure that there are no errors in replication or splitting of DNA, or sorry, splitting of the cells, to make sure, and the chromosomes and sister chromatids, to make sure that there are no issues in the organism. So for example, one main issue that you probably know happens when you have genetic malfunctions is cancer, or other genetic diseases, especially hereditary diseases, are caused by errors in mitosis. So to prevent this, we have a lot of stages, checkpoints, and mechanisms to kind of keep that in line. But prokaryotes don't really care. Obviously, they don't care if they get cancer because they can't really get cancer because it's just expanded growth or something like that. They don't care about genetic defects. So prokaryotic fission is, as a result, much, much faster, but it's also a little bit less accurate than eukaryotic mitosis, and has a lot less steps, a lot less going on. And here is a example of binary fission. You can see it's much simpler. It creates a copy of the DNA right here, right? The replicated DNA. Then it elongates, kind of like split more. Then it forms like a cross wall, and then it just splits apart, right? Very simple compared to mitosis, which has like the G phase, the M phase, and all of that. Okay. Okay, now moving on. As mentioned before, bacteria are the most common prokaryotes. They come in 3 distinct shapes. Remember, the other prokaryotes are archaea, but most of the prokaryotes that you'll see at least are bacteria. They come in 3 different shapes, and these shapes are rod-shaped, spiral-shaped, and spherical-shaped. And these have different names. Rod-shaped are called the bacilli, spiral-shaped are called spirally, and spherical are called kochi, I don't know. But yeah. And you can see here some examples, Streptococcus, right? That's very common ones. Some of these flagella rods, the Salmonella, H. pylori, yeah. Okay. Next is bacteria cell wall types. Mentioned all prokaryotes have cell walls, but there are 2 different types of cell walls bacteria can have: gram-positive or gram-negative. Gram-negative bacteria have a thin peptoglycan layer and outer lipid membrane. Gram-positive bacteria have a thick peptoglycan layer, and no outer lipid membrane, right? And the main distinction, so these are the 2 main distinctions in terms of what they actually are, like biologically. But actually, another way we distinguish gram-positive versus gram-negative is through like fluoresis. So when you do an experiment, gram-positive will be purplish, whereas gram-negative will be, I think, a bit like reddish. I'm not sure, but I know the color is a big difference, and I know gram-positive become purple. Okay. Okay, and this is kind of just an example of what they look like. So as you can see here, gram-positive have a thick peptoglycan layer, this little purple thing, right? It's very thick. There's a lot of layers, whereas this one in gram-negative is very thin. But gram-positive, there's nothing above this, but in gram-negative, they have this lipid layer, right? You can see here, this looks like just any membrane. Like here, you have the phospholipid bilayer. Same thing here, you have another just lipid bilayer, okay? Outside of the peptoglycans, okay? It's not like super important, but sometimes you may be asked gram-positive, gram-negative, so it's nice to know. Okay, lastly, I want to cover on movement of bacteria. And bacteria are made mobile by a structure called flagella, which I mentioned before. Flagella are like propellers on like a boat, which allow the cells to move in an aqueous environment, which means kind of like in liquid. They're rigid filaments, just like kind of like how your muscles are like rigid filaments. They can work together to rotate and propel the bacteria in a direction. Flagella respond to a number of outside stimuli like light, chemicals, etc. So if you have if you shine like light on it, they may move towards or away from the light. If you put chemicals like sodium or whatever other chemical they respond to near them, they may go towards or away from it, depending on whether they need or whatever. And this encourages them to either run towards or away from such stimulus, or to simply tumble in place. Right, you can see here that these little string-like flagella, if they rotate counterclockwise, they can, it's called running, and they can actually just like propel it forward. So they kind of like propel the thing forward, the bacteria. Whereas if you have this clockwise rotation of the flagella, they kind of tumble in place. They kind of just essentially spin in place. And this is to prevent, especially in like an aqueous solution, to prevent it from being moved by the medium or being moved in any other ways. Sorry, it just spins in place and like tumbles in place to avoid as much movement as possible because it wants to stay in the same position. And that's different from running, which is obviously going away towards something. Okay.